Well, Katrine, thank you so much for joining us for
Thanks
this
for having
series.
me.
We're trying to do a showcase of our entrepreneurs and our startups to really help educate our community. Let us start by introducing yourself. Can you tell us your full name and your current company,
Yes,
if you're keeping track?
of course. So I'm Katrine Bosley, and I've been in the biotech industry for over 30 years, which, yeah, it's a fair amount of time. But I'm currently CEO of DaCapo Brainscience, as well as being chairman of the board of another company called Arrakis Therapeutics. Um, I've helped build a number of companies with a number of different technologies and in different therapeutic areas. I was CEO of Editas Medicine, of Avila Therapeutics. I helped build AdNexus, and I actually spent quite a number of years at Biogen back in the day. I think the common theme among all of them is new science emerging from discovery and really getting ready to be translated into making medicines. And that time period where you've got to put shape and direction to it to really go from exciting science to something that looks like a therapeutic and send it on its path into the clinic, that's the stage where I keep coming back.
Excellent. And so you are a true serial entrepreneur in that sense.
It's happened that way, yes. It's
And
happened that way.
that's a profile that's very highly sought after. Certainly investors have the comfort of someone who has seen that and done that. Here we often work with first-time founders, which is also very cool and interesting. They bring this unbridled enthusiasm and energy. And we'll ask you some questions today
Sure.
that will make sure that we guide them in the right way.
Yeah.
Maybe we'll start out by giving you a chance to deliver a pitch to an imaginary investor in like 30 seconds or so.
Sure.
For DaCapo Brainscience.
Yeah.
Let's go.
All right. Well, DaCapo is focused on creating novel medicines to address progression in neurodegenerative diseases. And this is, I probably don't have to tell anybody about the need. Alzheimer's disease, Parkinson's disease, these are devastating, and we really don't have good medicines for them. But a couple of the long-standing gaps and challenges in discovery in this arena has been, can you use human data? The human data sets are small. It's hard to extract insights from those human data. And the preclinical model systems, poorly at best, represent what's going on in people. The platform we've created that our academic founders, Lorenz and Vic and Jen, created really does address these two issues, and through both computational approaches and really advanced iPSC, or induced pluripotent stem cell models. Between the two, we've been able to discover progression targets. In the whole field, there have only been 2 progression targets ever discovered. We've got dozens, of which we've prioritized 9 that we're now starting to dive in and really look at to create new medicines for Parkinson's disease initially, and with a plan to go to other neurodegenerative diseases over time.
Wonderful. That's a very concise description. And I have the benefit of having heard your pitch now more than once.
That's true.
So I have a little bit of an advantage over the audience. But help us clarify. You are using this analysis of markers that are correlated with progressive degeneration to come up with your own therapeutic targets. And then you'll be delivering or developing drugs against that.
Yes. I mean, at the end of the day, making medicines is the goal.
Yeah.
And to do that, you not only need the people we have as part of the team today, but we'll build the team over time to bring the additional drug development expertise in-house. We actually have our head of research, Warren Hurst, has built many molecules, taken them into the clinic. So we do have some of that around the table already. And I've been around that table myself a few times, but the ultimate goal is to make medicines, yes.
Talking about the size of your company, how many people do you have on the team now at DaCapo?
We were talking about this earlier. It's always a little bit of a tricky question because I can tell you how many employees.
Count them all in as if they were full-timers.
As if everybody were full-time, including some of our key fractional people. There's at least 13 or 14 people who are part of our team. And I mean part of our team. Now, some of them are spending a day a week or even a half a day a week. But all really, we view them as part of our team and integral to our success. There's no question about that.
And so a big part of drug development, drug invention is, of course, this teamwork. And you've of course, experienced and lived that. Tell us how DaCapo came into its life. Like, did you invent it? Did you meet with inventors? Did they recruit you? What's the story?
Well, so as I have to say, I'm not a scientist. I love hanging out with scientists, but I myself, I don't have an advanced degree. But, you know, when you've been around a lot of great scientists, you kind of have that sense of what is compelling and new and worth digging into. About a decade ago, I met Vic Khurana at a Keystone meeting. For those of you who know Keystone meetings, they're usually pretty focused, pretty high science in a ski location, because everybody goes skiing in the afternoon. And I just met Vic, and he was clearly really sharp and really creative and just a very interesting person. The kind of person you're like, I'm going to keep tabs on them because they're going to do something interesting. I don't know what it is. And he's a MD, PhD, classic. I'm looking at cells in the morning. I'm treating patients in the afternoon kind of individual. Anyway, dial forward some number of years and he DMs me on LinkedIn. And to be honest, I don't pay much attention to LinkedIn. It's just, I have too many other sources of messages coming at me. But every once in a while you pay attention and I'm like, I remember Vic. He was a smart guy. I will answer his DM. And he had two fellow academic collaborators, Jen Peng and Lorenz Studer, had been working collaboratively through a grant from a program called ASAP to do some really novel work in target discovery for Parkinson's disease. And so he pitched me on it. He wanted to start a company around it because he thought it was ready to kind of translate out of the lab. And I remember thinking, sounds super exciting. Oh, it sounds really young. Oh, God, you need some help, don't you? Okay, let me just, I thought maybe I could just advise a little bit, this or that. And honestly, it just evolved and grew from there. And they had a fourth founder, Matthew Peck, who had come from another field, but had jumped into this company as well. And you meet these people, and what always draws you in? Cool science, great people. I wanted to work with these people, and I also really felt this was a truly new approach to thinking about neurodegenerative disease. I'd wanted to work in neurodegenerative disease for a while. And I have to say, too, I really always end up with companies where, as I sometimes say, you're plowing the first furrow, right? So it's a very unclear path because you can't rely on somebody else having done it this way and that protocol and this template. You're really figuring it out every step of the way, which is, on the one hand, exhausting, but incredibly exhilarating. And it is incredibly satisfying as well.
Yeah, we feel that in your energy and in many other of our entrepreneurs' energy. You're describing how you're plowing new ground, but then explain the DaCapo name. I'm a musician, so DaCapo is not brand new. DaCapo is played again.
Well, basically. So for anybody who's either a musician or perhaps knows Latin, they will, or sometimes Italian, folks who know Italian will come to it. DaCapo means from the head. And in music, it's used as an abbreviation to go back to the beginning and often finish the music in a new way. And that's what we, in our aspiration, hope to do for people with neurodegenerative diseases is if their progression track was going to mean a certain amount of decline, can we take them back to the beginning and let them finish their music in a new way?
That's wonderful. That's a great
cannot take credit
ambition.
For it. And well, you want to have that inspiration in front of you, even if that's an incredibly hard goal. You got to keep the patient at the absolute front and center of what you're trying to do.
Yeah. So let's talk about the progression of the company from the earliest outreach of your co-founder to where you are today. What have you achieved? What are the funding milestones, scientific milestones until now?
Yeah. So
and maybe what are the obstacles that you had to overcome along the way?
There's no obstacles.
None at all.
So, I've built a few companies, and I think each one, the external context is a factor in how you shape it. Right? There are some environments that are much more capital permissive, others that are much more capital constrained. You could build a great company in either environment, but you do have to realize the laws of gravity do apply to you. So in the time period that we've been putting DaCapo together for these first couple of years, clearly it's a external environment that's been increasingly negative and challenging and difficult. For better, for worse, I've seen difficult environments before, and so it felt very familiar. And so one of the things that we've been pretty careful about from the beginning is to make sure we had really clear plans before we hired people, because you hire people, you're making a commitment to them. And so we actually spent about the first year volunteering our time for one, but also, really, what is it we're going to focus on? What is what does that plan forward look like? How does this go from what you guys have been doing in the academic laboratory to what we need to do to build a company? And so there was a lot of, we had offsites, we did a lot of Zoom meetings. This is also a lot of pandemic era stuff. It was a lot of planning. And so there was science continuing within the academic laboratories, but a lot of it was planning. How are we going to translate this out? We then were able to get some initial funding from angel investors, so individuals, not a venture fund, but individuals. And with that, the very first thing that we did was we tried to replicate what the academic folks had done. And I just, for anybody who is bringing work out of academia, I so strongly encourage them to do that first. And it's not because you should doubt the quality of your academic founders. It's not that at all. It's simply, it's science, it's hard, and a lot of times, more than half the time, it does not repeat.
Yeah.
And, maybe you investigate it and you figure out why it doesn't repeat and you can fix it. And other times it simply doesn't repeat, and then you should stop. In our case, it repeated, which was fabulous. There were a few things we found, like we need to tweak this or improve this, or what have you, change how it's done in order to let it be scalable. But the very first thing we did was we said, can we repeat some of these key experiments?
That is so important, and one of the key things that I think LabCentral does for people, it gives you the place where you can cheaply set up quickly, and hopefully you'll use it to replicate the academic foundation.
Yes.
That gives you the confidence to then build on it.
Yes.
Yeah, it's you said that so well.
And I will tell you, every time I pitch to investors, I have a confidence in what we're saying because I know it repeated, right? And there's just no substitute for that. And I've been involved with companies where stuff didn't repeat, and we spent a lot more money, and then we went back to say, can we actually replicate the original findings, and we couldn't.
And sometimes you can't, and you're 50 million into it. It's
we weren't that far, but it was still painful.
Been there, done that.
But the other key thing that we knew we needed to start early, because there's a real time to it that you couldn't shortcut, was establishing these very complicated, sophisticated cellular models. And that's a lot of what we do here at LabCentral. We're always in that tissue culture room. But these iPSC models take time, and you can't, there's nothing you can do to speed it up. You have to invest that time. And we're doing it with a really capable team. A key person came from one of our founders' laboratories, and so she brought with her some of those really well-honed skills and knowledge and experience, which was great. But establishing those models so that capability, because that's a capability we knew we couldn't access externally anywhere. And I think that's the other thing. There's something, you have to just, there's always a build by choice. What do we do internally? What do we do externally through a contract research organization or what have you? And we knew that these models do not exist anywhere else. And having them internally, have to build them, and it would be a real advantage to have them once we had, but it was going to take time. So planning, initial fundraising, beginning to put those models in place, replicating the academic work. And then the other key thing is team building, bringing in, and looking for a couple of the people who would be our key leaders, as well as obviously the small focus team that would execute on this work. And that was a big piece of last year as well. It's getting to this point where we are still pretty small, and we need to stay small for the moment because capital is still precious, but we have that critical mass.
How many different cell lines do you run? Are they all Parkinson's cell lines? Are they all from one patient? Or?
So it's not just about cell lines. It's also about, so when you think about iPSCs, right? You take cells and you turn them into an induced pluripotent stem cell, and you can differentiate into different cell types. And the cell types, they could, so we are also beginning to work with where the cells initially do come from a patient. And so your iPSC generated models come from a patient or healthy sources as well. And neurons, mature neurons as opposed to immature neurons. Aged neurons, it turns out aged neurons aren't like non-aged neurons. And the diseases we're dealing with are diseases of aging, greater age. And so it's very interesting because in those aged neurons, you see biology that you don't see in the mature but non-aged neurons. So you really do see a distinction, and you can uncover biology, target biology, in some of these aged models.
Are there specific neural or dopaminergic neurons that you work with?
Well, so then further you have, for example, multi-cell type cultures, right? So it's not just about the neurons. Like the neurons live in an environment with astrocytes and microglia, which are an inflammatory cell, and the way they interact with one another and signal to one another is another dimension of the biology that we're able to recapitulate in vitro in a way that lets us understand how targets and cells relate to one another.
I saw a very beautiful multicolored image in one of your slides.
That was...
That I would love to put on the wall somewhere.
We can help with that.
Yeah?
Absolutely.
That would be awesome.
Yeah, it is. And so there's a few things that are really interesting. So that is what we call the triculture system, right? So triculture, so 3 cell types.
So you have glial cells,
glial cells, astrocytes, and neurons.
Wonderful.
So glial cells be microglia. So you've got these, you've got the neuron itself, you've got the support cell, and you got the inflammatory cell, the brain specialist inflammatory cell. Part of what is important in making those models work is actually the ratio of the cells, and getting it to a point where it does give you a representation of human biology. And I think this is also where, if you think, dial out a little bit and you think about neuroscience research, so many of the classic model systems, be it worms or fish or flies or mice or whatever, their brains are not like our brains, and they're not great representatives of what's going on in the human brain. And so these human cellular models that look like the cell types that we want to impact in patients and people are a fairly new and really, really invaluable. They're a missing puzzle piece. We talked about earlier these long-standing gaps in preclinical model systems. This is an attempt to be able to fill one of those gaps.
Yeah, maybe the traditional animal testing will take less of an important role, right? Maybe you still do tox there, but... It's
funny. I think of it, because in a prior life I worked in human genetic diseases, and the same challenge there, right? The animal systems, maybe there's not even an animal homologue of the gene. If it does, maybe it doesn't do anything, so it's complicated. I think of it more like a jigsaw puzzle, right? And each puzzle piece gives you a bit of the picture, but you have to look at all of them collectively to say, this does or doesn't. Because what are you looking for ultimately? Is it a good reason to do an experiment in people? Right? You're gonna, not only are you gonna file this IND and hope the FDA lets you, but you need to be able to say to a patient, please let me experiment on you. That's what you're asking them to do. So do you have a sound rationale supported by data? There's not a formula for what those data need to look like. You're going to have some that's your pharmacology, and in a mouse or whatever, sure you'll do tox. You have to do all of that. But for the pharmacology part of it, you may be able to do something about target engagement. You're certainly going to learn about pharmacology, maybe your pharmacology, pharmacokinetic, pharmacodynamic relationship. There's probably some pretty important stuff you can learn in the furry test tube, if you will. The biology relevance needs to come, certainly from, maybe you can do something in animals, maybe not, depends on the target, your cellular models, but then also I think go back to the human animal. And could you look in clinical samples or go back to human databases to get a better understanding of the biological signal in those human data sets? I think that's something that, has been not very accessible up until now, because again, this is where some of these really recently emerging computational techniques bring a lot of power to the table to find signal in the noise of these small, sparse, heterogeneous human data sets. So I think you need to put all of those pieces together to say, hey, this is a good target. Our molecule is a good drug against that target. We think it's a reasonable and an ethical approach to say, let's test this in people.
In terms of evolving model systems for neurodegenerative diseases, do you think we'll get to... Now you're already doing triculture, right? That's mind-blowing even 10 years ago.
Yeah.
To what complexity will we get in the next 5 years?
That's a great question for Lorenz Studer, who's one of our founders, because literally this has been the journey of his academic career for, I don't know, probably 10 or 20 years. And I think that it will continue to evolve. I think that the question of, is there a point at which you're done? Probably not. But I think that, the question of how can you best represent human biology in an in vitro system so that you can efficiently look at a lot of different targets, a lot of different models. We can get a lot out of that today.
Mm-hmm.
And so we are doing that. But, you're not, there's never a we're done moment on those kinds of things.
So you mentioned in your introduction that you are finding novel targets, really, that are related and that you can now go after to slow the decline, for example, right?
We certainly hope. That is our goal. That is our aim.
Well,
yes.
then we have to have ambitions, right?
Yes.
So what modalities would you employ to go after them?
Yeah. So practical reality, particularly with neurodegenerative disease, is that we're going to put our thumb on the scale with respect to small molecules because, as you know, to access the central compartment, to access the CNS, which we would anticipate needing to be able to do for the kinds of targets we're looking at, many other modalities don't get there. That said, I'm going to broadly speak, and so we do look at targets. It's very nice if we see an enzyme in our target list, or there's one in our list that's a kinase. I'm like, ooh, we like the kinase, because we know what to do with it, right? There's a lot of tools out there. It looks more tractable, so that's great when we see that. We're gonna, if the biology is compelling, that's gonna boost it up on the list for sure. Now, if we see targets that if it required an antibody, that's something you can pretty readily do. There's a lot of great antibody generation companies out there. If it requires what I'll call a more advanced modality, so an ASO, an antisense oligonucleotide, or any kind of genetic medicine, whether it's gene augmentation or gene editing or something like that, or I would also put in that category a PROTAC or any kind of ACT. There's lots of ACTs out there. Those are all wonderful modalities, but they're still new enough that they are not turn the crank. They're differing levels of extremely difficulty, need a specialist, or artisanal and only a few groups do them. They're not broadly available. And so I think we would seek to collaborate, and I will say that, over the years I've built a few modality companies, and there's a time period when a technology is new that there's huge value in that modality being the modality specialist. But as that gets worked out and as you really come to understand how to do it, and if it really works in a good way, it should be kind of commoditized. It takes a very long time, but look at making antibodies today.
No, with antibodies.
There's a time when there was one company where you could go to do that, and then there were a couple, and over 20 odd years later, you got a lot. So if these modalities are really robust and against a lot of targets, they will become sort of commoditized over time. But the biology, if you bring the biology to the table, you are bringing at least half, if not more, of the value to the table. And so I think that, those companies that do those modalities could be excellent partners. It could be a really great way to move a program forward.
Now, you've built or been involved in many companies, small and large, different types of modalities. Are you still learning things
Every day?
DaCapo? What have you learned since you joined or started DaCapo?
Well, certainly one of the big dimensions here that's very new to me is the very heavy emphasis on the computational approaches, which is obviously, I talked about dealing with sparse and small data sets. I know everybody talks about machine learning these days, but if you think of machine learning as a field where, of course, we all see these large language models and all that stuff's kind of interesting, and we engage with that in different ways. But as a technical specialty, the real cutting edge tip of the spear is dealing with small data sets, because not just in our field, but there are other fields and other disciplines where there aren't big data sets and there won't be big data sets. If you think of just from a purely statistical standpoint, of course, one of the biggest problems is how do you know signal versus noise? You've got one signal point in that data set. If you had a bigger data set, would it be just random noise? How do you know? And so that you can imagine from a mathematical standpoint, a computational standpoint, this problem is a hard problem. And again, there are a number of fields and disciplines where this is a common problem. And our field is one of them, right? There's no time soon that you're going to have 10 million genomes and 10 million 5-year longitudinal clinical data sets for people with Parkinson's disease. It's not going to happen.
Right.
So, but human data is the most valuable raw material for extracting biological insights in human disease. So can we meet the data where it is with more advanced computational and mathematical techniques.
So this is really your first foray into computational biology.
In some way, I mean, the computational biology, all this, that's in the groundwater for anybody who's, go back to Vertex, right? I mean, that might have been the first company where Josh Boger had to convince people that using computers was useful in drug discovery, but like it's been kind of part and parcel of how everything gets done. But there are these moments where there are, I always like the analogy of a supersaturated solution that one more little bit and then it crystallizes, right? Where something matures enough in its own domain that it's now useful outside of its little area of expertise. And I think that's what we're seeing, for example, with machine learning is that's now mature enough that you can use it in a lot of different places. So I'm learning there. I'm certainly learning with respect to IPSCs. I've had some involvement with those before, but not as a central element of a platform. And so having that be a central element of the platform. And I don't do the cells. That is, I would destroy them. Deeply respectful of people who have these capabilities. But it's fascinating to see what's possible there. And then,
is this the smallest company or the earliest that you've joined a company?
I've been a board member of companies this size before, but I guess the earliest before maybe a company was a year too old and maybe 10 people, 12 people when I joined before.
Now, with your perspective of having been an executive in public companies and on boards of public companies and now running an 8-person or 10-person, somewhat bootstrapped, maybe not really, but pre-seed funded company at LabCentral. That's a very rare perspective that we have. What are some of the things that you want to share from your perspective with other early stage startup founders that we have here, who most of them are scientists? They don't have any business training. They don't know how the game is played on the drug development side yet. They are typically target specific or gene specific or protein experts. What can you tell them in 5 minutes?
Yeah. So, probably the first, second, and third thing I would say is that this community, this biotech community, LabCentral, but I think just writ large, is incredibly available to you to help. We have all been there, and frankly, we all root for each other's success. The only way I can help the people, or I can pay it back to the people who helped me over the years, is to pay it forward. And I have been helped by many people over the years. And so I think that, there's an infinite amount of knowledge. You'll never be expert in everything. That's the nature of what we do. It is the ultimate team sport. It is the ultimate team sport. And so keep that open-minded and curious about all the different dimensions of what you can do, and reach out to people. The worst they can do is say no, but many more than you might anticipate will say, sure, and be happy to, give you some thoughts or give you some contacts or, give you some critique or tell you why that's the too high of a pre-money or, tell you why, you at least listen to your pitch and tell you what I hear. So I think that do reach out and develop the concept of a kitchen cabinet, right? So whatever stage you're at, you've got your board, you've got your leadership team, but like think of the third leg of the stool. Develop just a small cadre of 3 or 4 or 5 people who, there will be times when, you obviously want to be transparent with your board and your team. It's not about hiding information, but there are times when you need to vent.
You need a sounding board.
And you don't want to vent to them, right? Or you need a fresh perspective who's not all in the same problems with you and just can pull your head out a little bit. And people you trust, and people are happy to do that. I think that's the thing, but develop that consciously. In my first CEO role, I remember when I started, I'm like equal measure excited and terrified. And, I'd read this book, What Got You Here Won't Get You There, which I actually recommend. It's pretty interesting. The title does basically say it all. But, I knew there were going to be all these dimensions to the role that I was completely ignorant of. And I had a good board, I had a good team, that's great. But I was like, all right, here are 5 people I know who have been CEOs who I could call when I'm panicked and I don't know what to do. And there were a couple moments when I just felt stuck. There's more not so much panic, but where I felt stuck. And I'm like, I don't know how to get through this issue. And they all called me back. They all said, like, okay, try this, or I once faced this situation. It seems they broke the logjam for me.
That's great.
And that is available to anybody. You have to reach out for it.
Yeah.
But it's there.
I think that's super valuable advice. I came to Boston not knowing anything about biotech.
Yeah.
And Boston is a very generous community and city where you can have this type of guidance available at your fingertips if you reach out, if you use it.
If you reach out.
Right?
And it's interesting because I've also heard people say who come to Boston from elsewhere that it's a hard community to break into.
I didn't have that experience.
It's just great to hear because, I mean, I moved here after college knowing nobody. I got my first job with a want ad in the Boston Globe. That's a newspaper. They used to have want ads in the newspaper. But if you reach out, you will get that. Get that engagement. I agree.
Yeah, that's really great advice. We don't need to add any more advice. If you just listen to Katrine and reach out to the network, right?
Which is vast.
This is a very optimistic industry. What are
you
most?
It's a job requirement.
What are you most excited about for life sciences, for our biotech sector for the next 5 and 10 years? What are you watching in terms of technologies or developments?
Well, it's actually at some level I voted with my feet, and it's the kind of stuff that I'm doing here. My career over the years, I'll spend sort of 5 to 7 years deeply in one world, and then I'll kind of move on to another world. In the in-between time periods, I'm always like tumbling and sensing what's going on. I think that that's frankly, that's how I found CRISPR. I still remember the first time somebody drew it on a whiteboard, and I was like, does that really work? Because that'd be cool if it did. So let's see, what's interesting right now? The big trends that you read about with respect to like large pharma sort of swinging maybe a little bit away from rare disease and back towards metabolic and neuro and things like that. You do want to stay aware of those things. I think those are important, but I've always been a bit of a contrarian, and I don't chase those trends, but sometimes you feel those trends catching up with you. I do think neuro is getting more interesting right now, and I think part of the reason is that you have some successes, right? Aducanumab or lecanemab, and some of the antibodies are maybe finally showing that it's possible to do something. Why is that important? So I remember way back many years ago when I was at Biogen, and the first molecules to treat multiple sclerosis came out. And multiple sclerosis had been viewed as a disease where you can't do anything. And I remember Al Sandrock saying to me, when I was told when I did my neurology training, don't even tell the patient the diagnosis because they're just going to be depressing and they can't do anything.
And that's very different today.
And here's the thing, the fact that you had a first couple of drugs work, right? So beta-seron and Avonex being the first two, and then there were a couple more. And for a long time, it was just those 4. But the fact that they worked made other people say, oh, I guess that disease is not intractable. Maybe I'll invest. So when you have something work, a lot more people say, oh, maybe you can do something there. So I do think that that's maybe a neurodegenerative disease, because MS is sort of also a bit of an inflammatory disease. But a neurodegenerative disease, with Lilly continuing to push forward and Biogen continuing to push forward, you do finally have a couple of cracks that I think have made a lot of other people say, okay.
I hope that we'll soon re-walk this experience with MS in the ALS field also, right? Now that we start to understand much better disease mechanisms and we can go and target it. I think also even for the dementia groups of diseases, I think I completely share your optimism there, I think.
Well, and I think in the dementia and progressive neurodegenerative diseases, the other thing I think is just totally fascinating are all the new ways of monitoring disease progress. Obviously activity monitors and things like this, but if you look at the classical clinical scores, which we can't completely get away from, because at the end of the day the regulators are going to demand them, it's very hard to establish a surrogate endpoint. However, a lot of things you're trying to figure out in an early trial, a 1B or a 2A kind of trial, do I see some kind of signal? Obviously, there's a molecular, is there some kind of biochemical signal, but do I see some other kind of, maybe it's not that very classical progression score, multi-component score, but is there something else that says I'm impacting the disease? And if it's something where a person can do it at home, you can do it every day or every week, so you get a lot more data points. And I think what's important too, you're washing away some of that variability, because when people do the scores where they go into the doctor's office, they're revved up for it, and is that performance representative?
Yeah, I think that everybody's wearing a Fitbit or an Apple Watch.
I got an old watch, but
you do. I appreciate that. But these things will capture so much data that we will be able to utilize for.
Yeah, and including things like,
scores.
for example, in Alzheimer's disease, sleep disruption is a big issue, and that becomes a bit of a downward cycle because you're sleep disrupted, and so then you perform less well cognitively, and so then and often too, for example, sleep disruption could be the issue that means a patient can't stay at home anymore because the caregivers can't handle that. That's when the person might turn on the stove or wander out the front door, and they just can't
deal with
manage the person in that home environment. Yeah. So what if you could handle those sorts of issues or better understand them? So I do think that's a really powerful wind at the back in neurodegenerative disease, even though there's a huge amount of work to make it real.
Yeah, but it's certainly going to be better in 5 and in 10 years than it's been 5 and 10 years ago. So that's a really easy projection to make.
Yeah.
We before we wrap up, we have an orange object here.
Yes.
And you know that at LabCentral we love the color orange.
It's a good color.
What does orange make you think of, or why did you think of this?
So, I'm a scuba diver.
Oh, wow.
And of course, one of the most entertaining fish underwater is the anemone fish, which most of us think of as a clownfish. They're very, very orange. Now, it turns out they're actually pretty common and easy to see, but one of the things about scuba diving is that the rest of the world literally evaporates and you are 100% in the moment. And there's few things that are more entertaining than watching an anemone fish. They can be pretty aggressive. They will come out and defend their little anemone for you. But it does make me think of being underwater and watching an anemone fish.
That's great. Focus, zoning out of distraction. That's great.
Yeah.
Well, Katrine, it's been so great to chat with you about this. We learned a lot. We wish you the best of luck for DaCapo.
Thank you. Appreciate that.
We will stay close to it. Let us know how LabCentral can support your team as you go through this.
Yes,
but also we evolve by hearing from our user base, like what works, what doesn't work, right? So thank you very much.
Great. Thank you. Pleasure.
Thanks.